Good morning, dear students of Class 12 Science. We are dealing with the chapter Strategies for Enhancement in Food Production, chapter number 9. So far, we have learned about animal husbandry and animal breeding in this chapter. So, we have completed animal breeding in the last class. So, now, in today's class, we will begin with the topic plant breeding. Okay, so the topic for the day is plant breeding. Now, can you remember the name of the scientist? Okay, Norman E. Borlaug. It's in the page number 513. Can you remember? It's in the page number 513. Norman E. Borlaug. B-O-R-L-A-U-G Borlaug. Okay, he has immensely contributed towards the revolution called Green Revolution regarding the production of food grains. Clear? So he was regarded as the father of Green Revolution. Remember, Norman E. Borlaug is regarded as the father of Green Revolution. And in India, now I'm confused. And in India, the credit for the Green Revolution goes to Dr. M.S. Swaminathan. Okay, so in India, we used to consider Dr. M.S. Swaminathan as the father of Green Revolution. Clear? So Dr. M.S. Swaminathan enabled the country not only to become self-sufficient in terms of food grain, but also to export the food grains to other countries. Clear? So this particular green revolutions was possible to a large extent mainly due to the adoption of new plant breeding technique or technologies. Clear? So new breeding technologies has been implemented regarding the production of large quantity with good quality of food grains. So that's why Dr. M.S. Swaminathan is regarded as the father of Green Revolution. Clear? So by the help of this technique, all right, by the help of these green revolutions, by the application of new technologies, a huge variety of, or let's say varieties with higher productions, higher qualities of plants like wheat, rice, maize has been produced. So this particular plants of food grains was not only able to produce food grains with higher quality or in higher quantities, but also they were disease resistant. Clear? So now we'll go through the definition of plant breeding and we'll see the objectives and the main steps of plant breeding. Okay, so first of all, let us talk about the definition of plant breeding. So what is plant breeding? Okay, so plant breeding is a technique of genotypic improvement. Right, we'll come afterwards. What do you mean by genotypic? So plant breeding is a technique of genotypic improvement of economically important crop plants to produce new crops that are better suited for the cultivation. It gives better yield and a disease resistance. So here the plant breeding, it's mainly focus on improving the genotypic, means the gene. All right, by improving the genes of the plant. For example, let's consider that if one plant, all right, is not able to produce higher quantity of food grains. Clear? Because we all know that a plant used to have a different variety, or used to have different species. So let us consider there are two species. All right, sorry, let's consider a plant which belong to the same species. So we have taken two plants. One plant which has got higher production, but it is not disease resistant. The another plant which belongs to the same species has got lower production, but it is disease resistant. So are you getting it? So one of the plant has got higher production, but it is not disease resistant. Similarly, the another plant which belongs to the same species has got disease resistant property, but has no production. So in this case, what the plant breeders can do? So over here, by using different types of technology, the plant breeders can combine the gene of the two plants. Alright, that means they are trying to improve the genotypic combinations of a plant. How? They used to combine the gene of the two plants so that the character of both the plants will go into the offspring. Now, what are those character? So from one plant we will take the gene that is involved in higher production, and from the another plant we will take the gene that has the property of disease resistance. So when we will combine these two particular genes with the desirable characters, then the hybrid offspring which will be produced contains the parental characteristics. That means that hybrid plant will contain the characteristics of disease resistant property and higher production property. Okay, so this is mainly the technique of plant breeding where we are trying to improve the genotypic character or the features of a economically important plant so that it can produce higher amount of food grains, a better quality of food grains with the disease resistant property, and so that it can be acclimatized to a particular environmental conditions or to a particular habitat. Clear? So classical plant breeding techniques involve crossing of pure lines. Pure line means those plants which will produce the particular character from generation to generation. For example, Mendel's pea plant. Let us consider the tall pea plant. So Mendel's tall pea plant must be able to produce the tall genotypic character from first generation, the second generation, and hence so on. Okay, so till the fourth, fifth, sixth generations, the tall character should be produced. That is called pure line. Okay, so classical plant breeding techniques involve the crossing of pure line with desirable characters. Now we can choose any type of characters which are desirable. So I can choose the plant with higher quality, okay, with good quality, with higher production, or with disease resistant. So this can be my characters, okay, which are desirable and that must be present in a plant, and I can select those plants with such desirable characters if they are present. Clear? Then after the selection of the parents with desirable characters, we have to go with their hybridization. Okay, we have to mix the two characters. Then after hybridizations, we have to go for artificial selection. Artificial selection means selection done by the human being. In nature, it's done by the nature, which is called the natural selection. But when we are going with the selection of the characters which are desirable, followed by the hybridizations, leading to the formation of offspring, then in the offspring, in the hybrid progeny, we have to go for artificial selection. That means those hybrid plant with both the parental characteristics must be selected. It must be done by human being only, so it is called artificial selection or man-made selection. Okay. After that, what we can do after the selection of the hybrid plant with the desirable characteristics, we can go with their multiplication. So by this way, improvement in crop plants are being carried out with the help of different types of molecular techniques. Okay, we have got the plant tissue culture also, which we'll learn afterwards. So by the help of different types of techniques, we are trying to improve the genotypic features of the plant, leading to their better production, better quality, and disease resistance property. Clear? So that's the definition of plant breeding. Now, what are the objectives of plant breeding? Okay, the first objective of plant breeding, or the aim of plant breeding, is to have the plant with high yield or the higher production. Okay, so there might be increase in the yield, and not only there should be increase in the production of the food grains, but the quality must be there. There must be improvement in terms of quality also. So improvement in the production and the quality of the food grain is the first objective of plant breeding program. Number two, disease resistance, or resistance to diseases and pest. So resistant to disease like, let's say, fungal disease, okay, bacterial disease, viral disease, and resistant to insect pest must be present. All right, so those plants which are produced by the plant breeding program, by the plant breeding technique, must be resistant to different types of viral disease, fungal disease, bacterial disease, and also they have to be resistant to the attack of the pest and the insects. Number three, early maturity. Okay, so short period of maturity reduces overall production cost. All right, and enables the farmer to raise an additional crop. So that is the third objective of plant breeding program. Number four is abiotic stresses or resistance. That means whatever the plants will be produced by the help of this technique, they must be tolerant. They must tolerate adverse environmental conditions. So whatever the hybrid progeny will be produced by the plant breeding technique, those progeny must be able to tolerate adverse environmental conditions such as salinity, okay, high salinity conditions, or let's say extreme temperatures, and even the drought conditions. They must be able to resist those abiotic resistance. Number five, better qualities. All right. So over here, what we are doing is that we are taking the desirable characteristics from the parents. We are going with their hybridizations, and we are going for artificial selections of the progeny with the parental characteristics. Alright, so that means the preferential acronomic characteristics such as plant height, branching, erect or trailing habit, and better nutritious, well as good, etc., must be produced by the help of the plant breeding program. That means the plant, whatever the progeny of the plants are produced by this technique, it must have better qualities. Okay, they must have better nutritional value. Clear? And last one is better adaptability. That means it must be able to adapt to new climatic conditions and new habitat or new area. So these are the objectives of plant breeding program, or the aims of the plant breeding program. Now we'll go through the many steps in the breeding, because plant breeding programs are carried out in a systematic way, all right, worldwide, in a government institutions and in private organizations. So there are different institutions who are carrying out this plant breeding program. So the following you can see over here in the figure in the screen, that these following are the few steps that are involved in plant breeding programs. The first step is germplasm collection for variability. We'll go through one by one, no need to worry. Number two is evaluation and selection of parents. Number three, cross hybridizations between selected parents. Number four step is selection and testing of superior recombinant. Number five, testing, release, and commercialization of new cultivars. And last step is multiplication of improved seeds. So these are the different steps which are involved in plant breeding technique or in plant breeding process. But today we'll only go through the germplasm collection of variability. We'll go through evaluation and selection of parents, and lastly we'll go through cross hybridizations between selected parents. Okay, we'll only go through the three steps or in today's class. Next process or next steps we'll deal in another class. Okay, so first step, germplasm collection and germplasm collection for variability. So before that we have to remember what is germplasm. All right, then we'll go with germplasm collection. So what do you mean by germplasm and what do you mean by germplasm collection? So germplasm means the sum total of all the alleles of the genes present in the existing and the past varieties of a crop and in the wild varieties, they constitute the germplasm. So over here, what do you mean by alleles? You have learned in class 10 what is allele. If you know the term gene, then you'll also have gone through the term called alleles. Alleles means the alternate form of a gene. So germplasm means the sum total of the collection of the allele of a genes. Alright, we have to collect all the alleles of our genes, not only present in the existing variety of the crop, but also present in the past varieties also, along with their wild relatives. Because whatever the plant today we are domesticating, they have been collected, they have been cultivated from their wild ancestor, from the wild varieties. Okay, so we have to cut all the alleles of our genes of the plant present in the present form, present time, in the past varieties of the crop, and also in their wild varieties, and that will form the germ plasm. Okay, and collection of all the plants and the seeds having all the diverse alleles of all the genes in a given crop is called germplasm collection. That means when we will collect all the alleles of our genes of the present varieties of the plant, the past varieties of the plant, and the wild varieties of the plant, then it will form the germplasm collection. So germplasm collection means collection of all the alleles. Okay, collection of plants and seeds having all the diverse alleles. So when we consider the sum total of the alleles, it is called the germplasm. But when we collect all the alleles of the plant and the seeds, then it will form the germplasm collection. Now the germplasm, it mainly consists of the alleles from all the cultivated improved varieties. Point number one. Number two, it contains the improved varieties that are no more in cultivation. Number three, it constitutes of or it contains old, local or destitute varieties. It contains the pure line produced by the plant breeders and wild species related to the crop species. So germplasm consists of such particular features of the plant and the seeds. Okay, now germplasm of wild varieties, okay, primitive races and exotic species is conserved to provide genes of different values to the plant breeders for better experiments. So when we used to go for the collection of germplasm, now that particular collections will provide a better tool for the plant breeders. All right. So germplasms, or let's say collection of germplasms, is one of the most important steps or the first step in the technique of plant breeding program. So first step must be the collection of germplasm, and it is the most important steps in the plant breeding program because it contains all the alleles of a genes of the existing varieties of the crop, the past varieties of the plant, and the wild varieties also. And in that particular collections we'll find all the cultivated improved varieties, we'll find the improved varieties that are no more in cultivations. All right, we are finding the old local or the desi varieties, we are finding the pure lines which are produced by the plant breeders. Not only that, we're also finding the wild species which are related to the present crop species. So germplasm collection is the most important steps and the first steps in the technical plant breeding program. Then there's important few terms over here. Kindly go to the page number 509. Okay, until right, go to the page number 509. Over there, kindly go through the term IBPGR. One more time, I'm saying that. Go through the word IBPGR. IBPGR means International Board of Plant Genetic Resources, because whatever the plant breeding program is being carried out at the present time, at the present scenario, it is carried out under the guidance of IBPGR. Okay, so IBPGR means International Board of Plant Genetic Resources, and this particular board will control, or they will take a look on the plant breeding program which is being carried out by different institutions which can be governmental or non-governmental. Okay, and germplasm collections are also carried out or also conduct live in seed, also seed bank. All right, and over here we have got two types of seeds. Kindly remember the two types of seeds: orthodox seeds, which are mainly kept at low temperature of -10 to -20 degrees Celsius, and they are under anaerobic conditions, means in the absence of oxygens. Then we have got recalcitrant seeds. Okay, recalcitrant seeds, which are also conserved in aerobic humid conditions at room temperatures. All right, and over here these particular seeds are mainly preserved at very low temperatures. Clear? These seeds are conserved at very low temperatures. Orthodox seeds were conserved as -10 to -20 degrees Celsius, but these Lactulent seeds are conserved at very low temperatures, around -196 degrees Celsius, which is called cryopreservation. Okay, so these recalcitrant seeds are preserved by means of cryopreservations. All right, temperature of more than -196 degree Celsius. So kindly go through this term. Okay, it's in the page number 509. Now, number two steps is evaluation and selection of parents. Now, germplasm collection has been done, which is the first steps of plant breeding program. Now, after the collection of germplasm, what can be done is that the germplasm which is being collected must be evaluated. Okay, it must be evaluated. Why? Because we have to select the plants with desirable characters. We have to identify the plants with the desirable characteristics. We have to find those alleles which bear the characters which we want to go for hybridization. So evaluation and selection of parents. So germplasm has been collected. Now go further evaluation. And during evaluations, we have to select the desirable characteristics. All right, so we have to identify those alleles with desirable characters and we have to identify them. Now after that, after evaluation, after finding out the desirable characteristics, we have to go for their selections. Okay, selection is carried out for plants with advantageous character based on phenotypic characters. Now what can be the advantageous characters? Let's say in terms of plants, it can be higher production. All right, higher production, better quality, disease resistance. It must be able to adapt to different climatic conditions. This can be the desirable characters which we can choose from the germplasm. So after the selection of parents, after the selection of the parents with suitable characteristics or the desirable characteristics, now those selected parents or the plants are grown under controlled conditions. Clear? And the seeds from the selected plants are harvested to reach the next generation. Clear? So what we have done, we have created the germplasm. Then after the formation of germplasm, we are going through the evaluations where we are trying to find out the plants or the seeds with desirable characteristics. I've given the examples. What can be the characteristics? Higher production, better quality, disease resistance, which can be grown in any climatic conditions, which can tolerate any environmental resistant factors like high salinity, okay, extreme temperature conditions. That can be our characters for selection. Okay, so after the selection of the parents, we have to grow them under controlled conditions, and we have to select the plants, sorry, the seeds from selected plants are harvested to mainly to raise them for the next generation. Okay, now selection of germplasms can be carried out in three levels. First one is mass selection, pure line selection, and third one is clonal selection. Just remember that the selections can be done in three ways. First one is mass selection, then number two pure line selection, and number three is clonal selection. Just remember this word. Okay, now after the evaluation and selection of parents, the next step is hybridization, cross hybridization between selected parents. Now what is hybridization? What do you mean by hybrid? What is hybrid? What in hybridizations? Hybrid means any offspring with the parental characteristics present in them. If the parental characteristics are present in a plant or in a seed, then that is called a hybrid progeny or a hybrid plant. So here what we are doing, we have selected the plant or the seeds. Now we are going through the hybridization, so that whenever the progeny or the offspring will be produced, then that offspring will carry the characteristics of both the parents. Okay, so we'll go through it now. Let's see. So hybridization is bringing together the gene pool of two different germ lines differing in their genetic composition. So what we are doing over here, just think of the example I've told right now. We have got two plants belonging to same species, all right, because we have got different varieties of plant or different species of plants. Okay, so one plant has got higher production but no disease resistance. Second plant has got no production but is disease resistant. So what we are doing, by the help of hybridizations, all right, we can combine the two genes, all right, the genes with higher productions and the gene with disease resistant property. Go for hybridizations. Now whenever these parental characters will undergo hybridizations, the progeny which will be produced will carry the gene or carry the characteristics from both the parents. That means the progeny will have the characters of disease resistance and the higher production character. Okay, so that is called the cross hybridization. Now over here, remember the term called line. A line represents a group of individuals or plants descended from the same or related parents. So through that line, we are going for the selection of the parents and we are going for the hybridizations. Clear? And the individuals or lines which are used in hybridizations, they are called the parents. Okay, very clear. So whatever the plants will be taken into considerations for hybridizations, they will know is the parental plant. Okay. Now over here, we are saying that whatever the characteristics that are present in the parent which are desirable, all right, that has to be inherited to the progeny. That means it has to be transmitted to the progeny. Now this is only possible by the help of cross hybridizations, which will produce the hybrid, all right. And now hybrid will be genetically different from the parental characteristics. Why? Because parental the characters or parental generations carry only one character, either higher production or disease resistant property, but the hybrid will contain two genetic characters. What are the characters? Number one, higher production characters, and number two, disease resistance character. And this is only possible by the help of cross hybridization technique. Now the process of hybridizations include two steps. All right, after we will go through the types also or the procedure also of hybridizations, but now let's go to the process which involve two steps. What steps? Pollen grains from the desirable plant chosen as male parent have to be collected and placed on the stigma of the flower selected as the female parent. Now over here, this question for you. What do you mean by pollen grain? Alright, because before going into hybridizations, you must be aware, you must have the knowledge about the male gamete and the female gamete of a flower, of a plant. So what is pollen grain? Pollen grain is mainly the male gamete of a flower or of a plant. Okay, so what we are doing over here, we have to take the pollen grain from the desirable plant. Clear? And after selecting the pollen grain with desirable characteristics, it has to be placed on the stigma. Now what is the function of stigma? If you remember, alright, we have got stigma, style and ovary. The function of stigma is to receive the pollen grain. So pollen grain with desirable characteristics, alright, will be selected, which is the male parental characteristics. Okay, now that pollen grains have to be placed on the stigma of the flower, which is selected as the female parent, because we have to select the male parent and the female parent, and during pollinations the pollen grain has to fall on the stigma and needs to germinate. All right, this thing you have done in class 9. Clear about pollinations. So what we have done over here, we have chosen the male parent and the female parent. From the male parent we are taking the pollen grain, and that pollen grain is being distributed or placed on the stigma of the female flower, which has been taken or chosen as the female parent. Clear? So pollination has been done. Now number 2 thing is that it is not necessary that the hybrid do combine the desirable characters. Usually only one in a few hundred to thousand crosses show the desirable combination, and such hybrid plants are selected. Clear? We are saying that if, let's say, we have gone for hybridizations, and during cross hybridization, let's say 100 progeny has been formed. Clear? So we are not sure that all the 100 progeny contains the desirable characters from both the parent, and that is also not true. One in a few hundred out of 100, either one or two will be hybrid. That means it will contain both the parental characteristics. Only one in 100 will become our hybrid progeny. Alright, or in the thousands, 100 will become the hybrid progeny. That means it will bear the desirable characters. That means the characters present in the mother and the father. Okay, that is the male parent and the female parent. So only one in 100 or 100 in a thousands will be the hybrid progeny. Now second step is the selection of such hybrid progeny. So we have to go for artificial selection. Okay, where we have to select such hybrid progeny having both the desirable characteristics from the parents. So there are two steps of hybridization. First one, we have to select the male parent and the female parent. After that, the pollen grain from the male parent will be placed on the stigma of the female parent which has been selected. Now we'll go for cross hybridizations. The hybrid progeny will be produced, and it is not true that all the progeny will be hybrid. One in 100 will be our hybrid progeny, or 100 in 1,000 will be our hybrid progeny. So we have to select those hybrid progeny which bears both the parental characteristics. Then we have to go for artificial selections. Clear? So these are the two steps of cross hybridization. Now let's go with the procedure of hybridization. First one, selection and isolation of plants. So first, the plant to be selected to be the male and female, alright, needs to be selected. First we have to select the male and the female parent. Okay, and both the parents, male parent and the female parents, are induced to flower at the same time, and crossing is done in a greenhouse under controlled conditions. So first step, first procedure is to go for the selection and isolation of plants. We have to select the male parent and the female parents. Clear? So first step is done. procedure. Second one is emasculation, which is very important. Now what is emasculation? So before going into the term emasculation, what do you mean by bisexual flowers? Clear? What is bisexual flowers? Bisexual flowers means those flowers which bears both androecium and gynoecium. Clear? So what is androecium is what is gynoecium? So you know it very well. All right. So bisexual flowers means those flowers which bears both reproductive organs, androecium and gynoeciums. Okay. So if the parents are bisexuals, in that case, stamens of one of the flowers are removed. Very important. Stamens of one of the flowers are removed. Now, what do you mean by stamens? What are stamens? Is it the male part of the flower or the female part of the flower? Stamens is the male part of the flower. All right. So if the parents are bisexuals, bisexual flowers, then in that case, what we are doing, we are removing the stamens. We are removing the anther of the flower. Okay, so removal of anther of a flower or stamens of the flower at the very young age or at the bud stage is known as emasculation. Okay, so second one, emasculation, where we are removing the anther or stamens at a very young stage or at the bud stage. If the flowers are bisexual, if the flower is unisexual, there is no need for emasculation. But if the flower is bisexual, then we have to go for emasculations, where we have to remove the stamens or the anthers of the flower. After emasculations, we have to go for bagging. Now, what do you mean by bagging? What we have done? The male part or stamens have been removed. Now there is only the female part of the flower. Now those flowers from which the emasculation has been done, has been carried out, needs to be covered with the polythene bag or the paper bag, so that no foreign pollen or the unwanted pollen may fall on the stigma. Okay, so when we used to cover that emasculated flower by the polythene bag or by the paper bag in order to avoid unwanted pollen grains to fall on the stigma, then that is called bagging. So first we are going for emasculation, then it is being carried out by bagging, where the emasculated flower is being covered by the polythene bag or by the paper bag to avoid unwanted pollen grain to fall on the stigma. Okay, so after bagging, we are going for tagging. All right, we have to write the proper every steps when carried out. We have to write the date and the time on the emasculated and the bagging flower. Okay, so we have gone for the tagging. After that, when the flowers will become mature, when it is ready to receive the pollen grain, bags will be opened. All right, and pollination will be done. Now over here, pollination will be done in such a way that the pollen grains from a desirable plant will be selected and will be dusted or will be placed on the stigma of a flower. Listen very carefully. All right, those male flowers which has been selected, from those flowers, from male flowers, we will take the pollen grain and we will place the pollen grain on the packed flower. Clear? Again, we will go for bagging. So these are the procedures which has been carried out during the process of cross hybridization. Clear? So today we'll only discuss the three steps. What we have done, we have learned the first steps was germplasm collection. Second was evaluation and selection of parents, and the third one is cross hybridization between selected parents. So the next steps, that is selection and testing of superior recombinant, then fifth one, testing, release and commercialization, and last one, multiplication of improved seeds. We'll discuss it in next class. Okay, so this much for today. So see you in next class. Thank you.